Good evening, everyone. My name is Scotty McLean and I am the Regional Agriculture Land Care Facilitator for the Limestone Coast Landscape Board. Welcome and thank you for joining us for our third webinar in the Healthy Soil series. I would like to acknowledge and respect the traditional owners of the ancestral lands of the Limestone Coast region. Potowitch to the north, Jarawajali to the east, Boandik to the south, and Miantung to the west. We acknowledge elders past and present and respect the deep feelings of attachment and relationship of Aboriginal peoples to country. So, tonight I would like to introduce you to Amanda Schapel. She is a Senior Consultant for Primary Industries and Region South Australia, or PIRSA, and has worked on agricultural soil since 1997. She started her career classifying and assessing soils for horticultural development in grapevines and almonds, and in the last decade has focused on the importance of organic carbon, particularly in sandy soils. Amanda completed her PhD in 2018, examining the relationship between clay concentration and distributor for carbon storage in clay amended or clay spread or delved sandy soils. She has been involved in a number of carbon projects, including the National Soil Carbon Research Program and benchmarking soil carbon in SA soils under a range of management practices and soil types. She is constantly learning new things about soil carbon and loves to share her findings. I will now hand over to Amanda. Thank you.
Hi everyone, and thank you for joining us tonight. So tonight we are talking about soil carbon and biology. And when we're actually thinking about soil carbon, we have to think about why it's important to us. And for people it will be different reasons, but the main option that we have really, or the main consideration that most people have, is talking about soil health and soil function. When we talk about soil health, Mel introduced the concept of the definition that the government is using, the South Australian government is using, as the ability of the soil to function as a living ecosystem in relation to its natural capacity. Really what it's saying is its natural capacity is those boundaries of your soil. So it's normally texture, so how much sand or clay you have in your soil, and often it's the mineralogy that determines its natural capacity. So it has a potential. And what we want to do is to get the potential of these soils to be working at its best. So a healthy soil sustains biological productivity. And the interesting part is that biological productivity is really one of the first times that we've had this in a lot of definitions. We're talking about maintaining environmental quality, promoting plant, animal and human health. And really it's resilient and profitable in agricultural systems. So when we're talking about boundaries for health and function, we are really talking about agricultural systems and what this means to us. And these boundaries that we have, or these definitions, are going to be different to what they would have been as a native or a natural soil, because we ask a lot more of our agricultural soils. So we need them to provide us with a function that helps us. And part of that is the plant productivity, so how we can grow quality food. So it's not always about growing more, it's about growing what is useful for us to feed our livestock, what is useful for us to get product off of, such as grain or grapes. We also want to know is the resilience of our systems. We seem to be coming up against more extreme events, and we really are looking for our soils to be able to bounce back or to come back quicker from these, such as the fires or the floods, the droughts, the frosts, and just being able to get going again in the shorter amount of time. And on my list I have down here, offsetting greenhouse gases is one of the lower ones. Greenhouse gases, soils, and storing carbon in our soils is really important, and it is a definite way that we can offset greenhouse gases. But if I put on my, it's not on the top of my criteria for when I think about why am I thinking about soil carbon and what's important. To me, it really is about the soil health and function, and if we can offset greenhouse gases, that is a bonus. So when we talk about soil carbon, we really are talking about soil organic matter. And what we forget is that organic matter is made up of not just carbon, but hydrogen, oxygen. And we also have the minerals such as the nutrients such as nitrogen, phosphorus, and sulfur. And it's in these parts that gives us our mineralization of our nutrients. Mel spoke about the different range of soil properties and differences that we have. Organic matter is critical for three components of our soil. We think of the physical, chemical, and biological components, and organic matter has a really strong part to play in all of these. So on physical, it helps determine the movement of water and air and the nutrient solution through for our plants. It's really important for a source of nutrients and a slow release form of nutrients. And it's important for cation exchange, for that nutrient of our positive cations, availability for our soils. And this is particularly more important in our sandy soils than our clay soils because it doesn't have clay to perform this function. So organic matter can improve our sandy soils, the cation exchange capacity there. We are also really interested in the biological form, and organic matter has such a critical role to play in this because it is the food source that enables the biological activity to occur, the diversity of our systems, and by doing this, we can also have improved suppression of soil-borne pathogens in our soils. So when we talk about carbon, we want to know what is it. So soil carbon is the part of organic matter that we can test in a lab. We have a test and we like to put numbers on things, and carbon is our proxy for measuring organic matter. Total carbon is of two parts. We have the inorganic carbon form, and the inorganic carbon is mineral based, and that's our carbonates in our soil. And anybody in the southeast is aware of what our carbonates are there. Some of those photos there show like a terra rossa soil, or you can have fine carbonates, you can have rocky in your carbonates. Whatever it is, the mineral based form that we have is not included in a lot of the carbon calculations that we have for ERF projects or carbon accounting projects. So the mineral base, the inorganic carbon, is not influenced by our land management practices, apart from when we put lime on for accounting for acidification. Organic carbon is really from plant, animal, or microbial origin, and it's living or decomposing forms of it. And we have organic compounds in there that can be quite complex. Now, organic carbon makes up about 40 to 60% of the mass of the soil organic matter, so it's a fair proportion of it, and it is definitely influenced by land management practices. So what we do has a great effect on what our organic carbon is. Now, organic carbon is made up of different fractions, or you can hear it spoken about as pools. And it's really important to know that the organisms, so the macro, meso, and micro that Mel introduced last time on the webinar, is really critical for the turnover of carbon and the release. And so this functioning in our soils that we really want to get out of them. So the different fractions that we have, there's 4 that are listed there. So plant residues that are greater than 2 mil. So plant residues can be above or below ground. Anything greater than 2 mil isn't included when we do a carbon test, a soil carbon test. So we're interested in anything that's below this 2 mil size. So it has to be sieved through and go through a 2 mil sieve. What we have is the larger fraction is the particulate carbon. You might also hear it called labile carbon, active carbon, or fresh carbon. And this is really the powerhouse source for the food for our microbes. And it's involved in this rapid cycling of nutrients and this availability of nutrients to our plants that we have. The size ranges from 0.05 to 2 millimeters in size. And this carbon stays within our soils for years. So we're thinking about a stability time of years. What's important to know about the carbon fractions is that continually they're turning over. So it's changing and it's dynamic. As we go from particulate form and we're transforming that, we're going into humus. And humus is the form that we are really looking for if we're looking for stable carbon in our soils. The size is less than 0.05 millimeters in size, and it's often associated with clay minerals. The residence time in this, we're talking about decades. So we're talking about a much more stable form of carbon. It is, it's harder for the microbes to break it down. Not saying that microbes can't break it down, because they definitely can work on these areas. The humus is really important for cation exchange capacity, as we mentioned before, for holding on to these cations, these nutrients in our soils, and for providing pH buffering and availability of nutrients to plants. Again, this is really important for sandy soils that don't have clays to perform this function. So more humus in sandy soils is really important for the functioning of our soils. And the last pool we have is the recalcitrant form. And in Australian soils, this really is dominated by charcoal. So anything that's happened from fire is called recalcitrant carbon. And this stays in our soils for centuries. It generally isn't broken down by microbes. So turnover is dynamic, as I said. And what we're finding sometimes is that we don't always see the fractions, the amount of humus or that we might expect from having a high amount of particulate. And what we're finding, so Clive Kirkby did some work and found out that nutrition can limit this turnover or this transformation of particulate carbon into humus carbon. And what he found was that to get about a tonne of humus, you need to have a proportion of nutrients, and that's roughly about 80 kilos of nitrogen, 20 kilos of phosphorus, and 14 kilos of sulfur. So the nutrition or the fertility or the composition of our soils can have quite a large effect on how these fractions are turning over. So what factors influence carbon? We have an upper level or a potential carbon that we can strive for, and this is largely determined by the soil type. And in reality, this is the texture, so the amount of sand or clay and silt in these soils. And from that, there is always the label that sand will have less carbon than what clay soils can have, only because of this a protection that clay affords carbon. Mineralogy also has an important effect, and the density or the mass of the soil determines the upper limit of what we can expect a soil to be able to hold carbon. We then have these other limiting factors, so we can step down, and from our potential, we now have what's called attainable carbon. This is affected really by the amount of solar radiation, so this affects how much photosynthesis is occurring. And we also have factors, and in Australia, rainfall is often our biggest one. Rainfall affects how much biomass we can grow and how much can be returned as a carbon input, but it also has an effect on the microbial activity. Microbes, as much as us, need to have moisture to be able to work. And in the same factor, temperature also affects not just the loss of carbon from direct loss of volatilisation of carbon from our soils, but also has an effect on what the microbial activity is too. So climate has a limiting factor. Then we step down again to what we actually measure in our carbon soils. And so this is our last reducing factor, and this is the management practices that we are using on our places. So there is a theory of carbon increase by management zones. And really the concept is, as you step up the ladder here, going from a cropping system to a pasture system, that our carbon values should be increasing. So what we started, the lowest that we could possibly have is a bare cultivated soil that has the lowest probably carbon values that we would expect. And as we start introducing measures such as minimum or no tillage and stubble retention, we start to be able to gather more carbon. And as we go up to introducing or changing over from a cropping to a pasture system, this is meant to be the maximum carbon that we can attain. There's also an option to add external sources of carbon to increase our carbon inputs into these soils if we can't manage it within our systems to be able to push them. It's interesting though, these theories of increase, as we're benchmarking soils in South Australia, we're finding that this rhyme or reason, it doesn't always work. So within some of these practices, we can have much higher or much lower carbon than you would expect from this theoretical increase. And this is where the benchmarking process is becoming to go really interesting. How do we get carbon into the soil? So really it's about the plant residue that we grow above, so the shoots and the roots below ground. These will form into carbon inputs that can go in. We also have sugars or carbon sources that happen via photosynthesis. So we get carbon dioxide from the atmosphere. It gets transformed into sugars that is exuded through the roots, and this is what Christine Jones has called the liquid carbon pathway. If you have livestock, you also have contributions from manure and urine, and you'll find that microbes themselves can make up about 1 to 5% of carbon that we can measure. And of course, being Australian systems, we always have charcoal or pyrogenic carbon, recalcitrant carbon in our systems. But what we have to remember is that with the carbon inputs that we have, we also start then have biological activity in our soils. And the activity of this biology starts to decompose the carbon that we have, and our losses can be of between 70 to 90% of the carbon that we're actually inputting into the soil. So how do we get carbon to stay in the soil? As we were saying before, soil has a limited capacity to protect carbon from decomposition, and this is largely determined by the amount of clay that we have in our soils. Organic carbon can bond onto the clay and protect it from decomposition by microbes, and there are also chemical complexes with iron, aluminium and calcium that can also protect the carbon from decomposition by microbes. The other option is to be occluded or to be bound up in these aggregates that we have. So when we have good structure, we have micro and macro aggregates. The carbon itself can be protected from microbial decomposition within the aggregates. If we have a lot of free carbon, and we're thinking probably sandy soils here where we don't have a lot of either the chemical or the clay complexing or the binding that we can have, we can be building carbon, but the risk of decomposition is quite high in these soils. So carbon can be stabilised by clay minerals and within aggregates. So when we actually take a soil carbon sample, we need to have some good thought about taking a good sample to make sure that what we are collecting is representative of the paddock or the area that we're focused on. When we take samples, we often take a sample, so same with an agronomic sample, in a 0 to 10 centimetre if in a cropping system, or a 0 to 15 if you're pasture or viticulture. And what we do is we look at the number of samples what we can have. This graph here that you can see on the left shows the number of samples down there on the x-axis, and it shows the soil organic carbon value on the y-axis there. And then you can see there's a dotted line at around about 10 number of samples. And so what this is basically showing is that below 10 samples, you can see that the carbon values will fluctuate wildly. So the confidence that you would have in a sample where you have collected less than 10 samples will be quite low, only because of the natural variability in that area hasn't been captured. As soon as you get above the number of samples, about 10, you can see that fluctuation decreases and it becomes much more steady. So you have a much greater confidence of samples when we're going above 10. And those samples there that we were looking at there was from a sandy soil, and it was in a 25 by 25 metre grid. So that's the size that we use for a lot of our long-term monitoring sites, where we go back and remonitor every 5 to 10 years. If you were doing it in a paddock, you would look at taking a minimum of 20 samples, and you would try as much as possible and section your paddock up into textures, because as we said before, the amount of sand or clay in your sample has a wide effect on what your carbon will be. Other options that we're looking at too is we're focused a lot on the 0 to 10 centimetre sample, but there's quite a large opportunity we're finding to increase carbon below 10 centimetres. Often between the 10 to 30 centimetres, we can have quite an influence on what the carbon content can be. So it's really important to start collecting some samples now below that 10 centimetres. What we've often found is that the biological activity is quite strident, is quite large in the 0 to 10 and often the 10 to 20 centimetres. But below 20 centimetres, you can often take half the number of samples of what you would be doing in your topsoil. So if you were collecting 20 samples in your topsoil for your 0 to 10 or your 10 to 20 centimetres, below 20 centimetres, you could look at collecting just 10 samples in there to get a good representation of that natural variability. And that's often because the microbial activity is lower in the 20 to 30 centimetres, so that variability is a lot less. The other thing to consider is when you take your samples. So within a year, carbon will fluctuate, and this is dependent on what biomass is there, what the temperatures are, what the moisture is. So the important thing to do is to pick a time and then to stick with that. So if you're sampling every 3 to 5 years, you try and pick the same time. In cropping, we often say it's after harvest or in summer, and that's largely because there's little active root growth and the temperature is hot and the moisture is low. So the stability of that carbon is staying there and it's quite consistent. When we're talking about a pasture system though, you don't have to be as exact about that summer period. You can take it at any time, but what you would like to do is take it at the same time. So if you select sampling in autumn, you would then forever, or for successive periods, you would sample again in autumn. You don't want to get changes in carbon just because we've managed to measure it at a different time. So you've collected this fantastic soil sample. Now you go to the lab websites and you find that there are over 20 to 30 different tests now available for testing carbon. And and this can get quite confusing. The first test that most people are aware of is the organic carbon test. So the typical agronomic test that we often get in the 0 to 10 centimetres has this walk clear black method. And we still think in South Australia that this is a great test for us. And this is really because it's really important in calcareous soils because it doesn't measure that inorganic carbon or that carbonate in our soils. The only problem though is that it's called an incomplete carbon test, organic carbon test. It only measures 55 to 90% of the total organic carbon. So some labs will offer up this next option, the total organic carbon, and it's called a Heinz method. So they apply a heat to it and they get what would be a complete organic carbon or total organic carbon test. The national standard that is going to be adopted or has largely been adopted now is to have a total carbon test. And this is via high temperature combustion called the Dumas method. You can often hear it called a LECO test, and LECO is the name of the machine that they use to analyse the results. High temperature, and most of these are combustion at just under 1,000 degrees. The only issue that we have in soils, so for in South Australia where we have no carbon in our soils, this is a great test. Unfortunately though, if you have carbonate in your samples, it does measure the organic carbon and the inorganic carbon component as well. This probably isn't an issue if you only have small amounts of carbonate in your soil, but as soon as you start to have moderate or high amounts of carbonate in your soil, it can be very difficult to detect a change in organic carbon because of that large influence of the inorganic carbon. So the option there is to then look at putting a value on the inorganic carbon test. Now, a lot of labs in the past have done the calcium carbonate equivalent, and they have tried to calculate the carbon based on calcium carbonate and do a calculation. The only problem is it is often over calculated the carbon in the calcium carbonate, and you're left with the total organic carbon value very low. The preferred treatment now, and the majority of labs are offering this, and I know AgPower is about ready to offer this commercially in the next couple of months, is an acid pre-treatment. So basically they add acid, they remove all of the carbonate from the soil, and then they test it through the LECO machine. And this is definitely a much more accurate test for us with calcareous soils about getting a really good total organic carbon value. Just be aware though that we've been sending samples off for different labs, and different labs have different methods of how much acid to add to the carbonate and how many times they will do it to remove it. So once you pick a lab for testing carbon, stick to it, because there are slight minor changes within some methodologies that can make a difference when we're talking about very small amounts sometimes in our soils. The other test of interest is the mid-infrared spectroscopy. It is becoming more and more popular. Lots more labs, the commercial labs are offering it. It's relatively cheap, but it's not as accurate as the other methods that we're speaking about. And it does rely on having a very well calibrated data set. The bonus about this when we get more samples through is that you will be able to get your fractions. So that particulate and humic fraction, you'll start to understand how much they comprise in your soils. Now there's some biological options here, and I have to say I'm definitely not an expert in biology. This is just an area that I'm only just touching the tip of the iceberg on. And so I've adapted a slide that Mel had from Helen Hayden, so from Ag Victoria, about biological options that we can use. And really she's separated it into assessments, visual assessments that can happen on farm that can be done by managers. And this is looking at species abundance and diversities of things like earthworms. Springtails are becoming really quite accurate at measuring them. We've also can look at ground cover, so your composition of your species and also the amount of bare ground that is there. Important things like topsoil depth, the rooting depth that you have. Don't just look at rooting depth, but also look at abundance, so the number of roots that you have and the health, the score of what your health could be for these areas too. These can be done very easily, quite cheaply yourself, and you can do repeat assessments. But again, just be mindful that you're assessing them again in 1 to 5 years' time at the same time that you did your original assessment. You can also get tests that we were talking about with from your commercial labs where you'd get your soil tests. These are the soil carbon tests that we just spoke about before. There's another test called labile carbon, which uses a potassium permanganate version. This is popular particularly internationally. There's also a test potentially mineralizable nitrogen, which lets you know the capacity of your soil of how much nitrogen is available due to biological activity. Then you move down into some of these specialized biology tests. And here we're looking at getting measures of the first one, the microbial biomass carbon and nitrogen and the carbon dioxide respiration. They will give you a general feel about how much carbon and nitrogen is in the microbial biomass and also the biological activity of your soil. So you get a general overview, but you won't get any detailed information. When you go down to the next step and you're using tests such as enzyme assays, you're looking for more particular functionings of that you have in your soil. So you're looking for enzymes that particular groups of microbes use, such as cellulose or phosphatase to break down phosphorus. And then we step down into the really detailed things. So we're talking about looking at individual species groups or species within these that we have, so functional groups and species. And these can be like direct microscopy. We can look at DNA tests, looking at the soil microbiome, getting a lot more information. And we can also have a test such as pathogens that a Predictor-B test that SARDI offer. So we're really lucky in South Australia that we actually have 3 labs that offer a range of these different tests. So the Soil Food Web Institute, which is based at Millicent, with Chris Ellery, looking at the direct microscopy. Sorry, it's a word I'm having a hard time with. The Microbiology Laboratories Australia is based in Adelaide, and SARDI is based at the Waite there in Adelaide as well. These tests will give you a lot more information than the above tests. The part that will come will be the interpretation. We are involved in quite a number of projects at the moment, trying to gather a lot of data, a lot of tests, and trying to get meaning about what these are, what tests are good for growers to use, and at what time. So in the next year or year and a half, we should have a lot more information about what suits South Australian soils. When we talk about reporting carbon, when we get our carbon results back from our soil tests, we get them generally reported as a concentration. So we have it as a percentage. So if you have 1.5% carbon in your soil, you're looking at 15 grams of carbon per kilogram of soil. When we're talking about carbon accounting though, they're talking about carbon as a stock. So it's a mass or in tonnes of carbon per hectare. To be able to calculate that, you also need to have the bulk density or the mass of the soil of the area that you're looking at and the gravel content. And there are calculations that they use to be able to measure this, to be able to calculate. What we have then is stock is often considered in the top 30 centimetres, so not just the top 10 that we're often used to looking at. So you have carbon stock down to 30 centimetres. And you can convert your carbon, your tonnes of carbon per hectare, to carbon dioxide equivalents. And to do this, you multiply your carbon tonne factor by 3.67, and that will give you the carbon dioxide equivalents. So what do we know about soil carbon in South Australia? We've got a number of projects. So we've been lucky enough to work in collaboration with the Sustainable Soils groups between Department of Environment and Water and PIRSA probably for the last 3 years. We've been concentrating on gathering as much data as possible to understand what we know about our soils in South Australia and the opportunity, the areas of opportunity to increase carbon storage. So this report has been published now and was released last week. And I've got at the end, I've got the website where you can download this information. And what this report is pulling together is some benchmarks. We're providing benchmarks for farmers to be able to assess where their carbon values are. And to do this, we were able to, when the state government used to have a soil lab, we used to be able to pull together the data results of soil tests. So what we were able to do is analyse those that had organic carbon, and this was using the Walkley-Black method, that had that recorded for different soil textures, for different land uses, and for different areas. So we were given data that was done on postcode, and the land use was recorded by what was done on the form, and the textures range from sand up to clay. And so we ended up with 36,000 soil test results that we could do a general trend analysis of. Now, this is by no means perfect. We know having soil test results and how they were collected isn't always the perfect measure, but it does give us quite a strong indication about what is happening over time. The grey line there that you can see is the average of all of the soils, the 36,000 soil test results, and we can see that over time, organic carbon has increased by 0.08% per year from 1990 to 2007. When we look at these by land use, we can see the green line there is pasture, and that increases at a slightly higher rate of 0.11% per year. And if we go down to cropping, that blue line down the bottom there, we can see that it's smaller, 0.04% per year. But the good news story is it's a positive trend. We often hear that our agricultural systems are decreasing soil carbon. And that is definitely been true from past history. From when we had converted the native soil, we did get a decrease in carbon. But from the management practices that we have been installing since 1990, we can show a slight general increase in carbon over time, up until the point of 2007. Unfortunately, we don't have any data as yet from 2007 to now about what has been happening since then. But if we go by and use these values, we can show some kind of positive trend. And also there, I forgot to mention that the orange dots there shows the orchard and vineyard. And as you can see there, the variability is quite large. And this would have to do with a lot about the vineyard boom that happened in the 1990s, late 90s, and about sampling. And we're actually looking at this orchard vineyard soils in more detail in the next year. What we can tell too is look at soil texture by land use. So here we have cropping on the left and pasture on the right. On the y-axis, we have the organic carbon, and again measured with Walkley-Black method. And down the bottom there, we have soil texture. So we have increasing values from sand to the left to clay to the right. Those black numbers that you see are actually the carbon averages of that black line. So that's the average or the mean line. The red line is where we found the 25th percentile of results, and we're using that as our lower limit. And the green line is our 75th percentile or our upper limit. And we'll set the 75th percentile as the target for where we would like to move if we're looking at modelling, where we move carbon values to. Notice on both of those there is also a grey line, and that is the state average. And what you can see, the state average in the cropping site sits on the 75th percentile. So the values there are around the 75th percentile. When we go over to pasture, the grey line or the state line there sits on the 25th percentile. So pasture soils generally have values above the state average, whereas the cropping have below. This is largely a result of rainfall. So when we're talking about what we see, rainfall has a large influence on what the land use would be at the time. When we have a look at both those areas, we see that you do see an inclining trend of carbon by texture. And we know this as we have more clay in our soils, we can have more protection of carbon. That potential gets higher to be able to store more carbon. And we also see there that the bands, so the difference between the 75th and 25th percentile is a lot narrower in cropping soils than it is in pasture soils. And again, we think this is largely related to rainfall and the amount of biomass that can be grown. And the target would be for our cropping soils is to gradually lift that 75th percentile up with different implementation of management practices. As we said, the effect of rainfall on carbon in South Australian soils, we're finding that we've had two different studies where we've looked at what the carbon values are dependent on rainfall. This one shows again on the y-axis your carbon in the Walkley Black content. Down the bottom there we have rainfall values to 250, 350, etc., grouped by clay concentration. So basically again on the left-hand side we have sands going up to clays. That little red line there often is that peak where you start to see the carbon value shoot up, and that's often around that 550, 600 mil. So we found overall in state averages, the average carbon, organic carbon, did not go above 2% unless rainfall was greater than 600 mil annual rainfall or about 500 mil in your growing season. And this is important for us because often when people talking about benchmarks for organic carbon, 2% is a value that is used as a healthy soil. And so we can see that in some of our systems, it can struggle to get to these values unless you have the rainfall there. So with this in mind, we have within that report, we've broken down the carbon benchmarks into agricultural districts. And these agricultural districts are based on the PIRSA cropping districts. And for each area then, we were able to look at these carbon values broken down by land use. So for the lower southeast there, we have pasture of 57%, cropping of 28%. And the little green area there, the mean value for the ag district was 2.47%. And this is much higher than the state average zone, the ag zone average of 1.77%. We also can then look at the annual change in soil organic carbon. And what we see in the lower southeast is a trend that's perhaps going down. Anybody who does stats knows that those R-squared values there are not statistically significant. So we're not saying that this in any way is a statistical study, but it's just showing us trends about what's happening. You can see there is a wide fluctuation in carbon values during those years over that time. And so this would have a lot of contribution about these trends. But in the lower southeast, we either have a very stable, very slowly declining trend in change in carbon. And it's interesting to talk to people about what these values could be. So it's been suggested that it may have been the changeover from pasture to cropping soils. It may be, there may be a lot of other reasons. So we will be doing some more benchmarking in the southeast a little bit later on, and hopefully we'll be able to understand some of these. The beauty about this really though is these ag district benchmarks by texture. So if you're in the lower southeast and you have your carbon test result for, these are all 0 to 10 centimetres, you can get your value, your carbon concentration, and you can compare it to what your value would be for that texture. You can then get an idea of where you might sit in the benchmarks for these areas. Here we've got the upper southeast results. So we have cropping, so it's switched over. We've got a predominant soil test results from cropping is 54% and pasture is 31%. You notice that the ag district here is 1.45, so it's lower than the state ag zone average. And that really has to do with that amount of cropping rather than pasture. Here we have a more steady annual change in carbon of 0.01%. So really it's either stable, I'm not sure we'd call that slightly inclining. But again, we have those ag district benchmarks where you can start to get an idea of where your carbon values fit for your paddocks within the texture of there. What we're doing for bigger projects is trying to get an idea of identifying carbon opportunity. So we're using these values for the areas or where we've collected a lot of benchmark values. And we're using this 75th percentile as our target. So you get your carbon concentration for the area, you divide it by that 75th percentile, and you change that into a percent. And by doing this, we can then start graphing these areas. So here we have organic carbon Walkley Black, so it's the value there. And then down the bottom there, we have that percent reached of that 75th percentile target. So for any site that is 100% and above, this means that it's hit that 75th percentile of the benchmark target that we had, and the opportunity to increase carbon further is limited. I won't say it's not there because we can always tweak systems and we can always get it to go higher, but these soils probably aren't your priority areas if you're looking for an area, a place to start. We're increasing. What we are looking at is probably those soils that, those blocks or those paddocks that sit below the 80% of areas that have that high to moderate opportunity to increase carbon based on those benchmarks that we have. And it just gives a bit of a starting position for where you might like to be. So now we go back to thinking about why we're interested in carbon. So a lot of interest that I've had, a lot of questions that we've had has been about joining carbon accounting or ERF projects or voluntary schemes that we have. And that's all well and good. As I said, the climate mitigation is something that's important. But the focus of just building carbon, when we start to focus on just doing one thing, we often have unintended consequences of others areas that we have in our systems. So increasing carbon over time can definitely occur. What we need to realise is that as we have our organic carbon inputs, we're going to be building our biology. So carbon won't increase linearly. It will come to an equilibrium. And at each time that we hit these equilibrium points, so where our outputs and our inputs are roughly matched again, we'll have to introduce a new management practice or do something different to set the line going into a linear mark again. So that's something that needs consideration. When we're looking at and focusing on increasing carbon over time, we need to also consider, will this increase function? Are we getting the best function out of our soils? If we go to the other side and we're looking about increasing soil function or soil health, and we're focusing on all of our inputs that we can have, and it's turning over really fantastic. We've got this biological activity that's giving us what we need out of our soils, and our fractions are all in balance where we need them to be. Our inputs and our outputs are balanced again. So will this increase carbon? And this is often what we're finding in some of these sites where we've thought that a management practice is going to increase carbon, and yet we haven't measured it as yet. It could be that we need longer times. The lag time for carbon can be anything from 3 to 5 years, up to 10 years, depending on what values you're talking about. How long have we got to wait, and how patient do we need to be? So we need to really, in everybody's idea, just have an idea of why we're interested in carbon and what our option is of doing this. So when we're talking about increasing carbon, we're thinking about the opportunity to increase carbon, and we go back to the capacity of the soil to store and protect more carbon. So again, this goes back to what the texture, that potential of your soil is, the mineralogy, the texture. You can use those benchmarks to get you an idea of where you sit on the scale of things. You also need to ensure that your carbon inputs are more than your carbon outputs. And we're really talking about decomposition and erosion here, and tweaking the system. I mean, that they're talking about having a different diversity, more biological diversity is to have more of an efficiency of breaking down carbon is supposed to decrease the amount of decomposition that we have in our soils. I haven't had any data and we haven't recorded anything different in that, but it would be an interesting process to follow. We also have to think where we have the possibility within our systems to increase carbon, that we are able to maintain carbon. And this is particularly true if you're talking about entering into a carbon accounting project, where if it's an ERF project, you're probably looking at a 25-year project that you have to maintain that carbon value at that site for 25 years. And we also have to think about having sufficient nutrients in our soils to be able to allow the transformation of the particulate fraction to the humus fraction, so we can have this longer term storage of soil. So we're looking at that decade storage in our soils. Now, these are some options how to build soil carbon. This is definitely not an exhaustive list. If I was looking at doing it, would probably have something that are 5 to 10 pages. So these are just some common practices that we can use. So really it's about minimising those losses, and particularly it can be about minimising erosion. So looking at cultivation practices, grazing management, and it's all about keeping cover on your soil, so reducing the amount of bare soil that we have, and for keeping cover as long as possible, and often for keeping green cover as long as possible. And thinking about species mixes and diversity can help you work these out. Also looking at maximising these organic matter inputs, so about growing biomass above and particularly below ground. So getting roots deeper into the soil, looking at the root architecture, the structure, the depth, the plant functions or the functional groups that we have growing in these areas can help start pushing carbon deeper into the soil through the physical roots and also the root exudates. Also looking to understand our soils and addressing or assessing if we have any constraints to production, anything that could stop this above or below ground biomass production. And these are things like pH or compaction. It could be sodicity, it could be a range of other things. And we're looking at nutrition targeted to production, really trying not to have excess nutrients available. We're looking at grazing management to encourage the exudates, the root exudates, for trying to push the roots deeper and for getting as much sugars in as possible. But we're also doing this within a framework of looking that the practices are profitable and sustainable over time. And really probably the biggest thing that we're going to be doing is learning and adapting as new information comes along. We really are at the start of something here. So the next 1 to 5 years is probably going to come along with a lot of information and a lot more data. So these are the references that I spoke about in there. So if you want to take a screenshot, or otherwise these can all be available, and I'll send the ones that I have options to. So the health check for our agricultural soils is just talking about healthy soils. That's a fact sheet. The soil carbon, the benchmarks and data analysis, I can send a copy to Skylee if she needs. Or you can download them from that Land Resources home website there. And if you just look under the all reports, you can download them yourself. The Soil Health, Soil Biology book there is a really, really good resource. And that's written by Graham Sterling, Helen Hayden and others, and it has some fantastic information in there. And Department of Ag Victoria also has some fantastic information, detailed information on biological tests if you want to look at it further.